Hello guys and welcome back to Excel Sheet Academy. So in the previous lecture, we discussed about the cyclin dependent kinases and the cyclins, right? So if you remember CDK4, CDK6, they bind with cyclin D, then CDK2 with a cyclin E, right? So that's how we enter the S phase. Now let's discuss more in detail about the S phase and the regulation of DNA replication. So CDK2 cyclin E complex, as now we know that it initiates S phase by activating DNA synthesis at replication origins, right? And we know that in the case of the mammalian cells, there are thousands of replication origins, right? So the initiation of replication at each of these origins must be carefully controlled. So each segment of the genome is replicated once and only once during the S phase of the cell cycle. So this also we know, like during the replication, DNA replication, we have talked about it, that in the case of the eukaryotes, many origins of replications are there and each origins are regulated. It means to ensure that in such a, it is regulated in such a way that in the cell only once the cell cycle or once the replication will take place, and after that it should be followed by the mitosis, and then again the cell cycle should repeat. Now DNA replication is initiated by the activity of MCM, that is mini chromosome maintenance helicase proteins, which is regulated by CDK cycling complexes at different stages of the cell cycle. So ये भी हमने discuss किया था when we were talking about the DNA replication. So first thing is that the MCM helicase proteins, they come and bind at the origin, okay, together with the origin recognition complex during the G1. So आपका क्या हुआ? If you remember DNA, if this is the origin, here your MCM will come and here ORC will come. Okay, let's see. I think I have the figure also. So DNA replication. Oh, 1 second. Sorry for this. Yeah. So DNA replication, as you see here, we have got the origin of replication. Okay, let's say that this is origin of replication. This is in the G1. Now, ORC comes and binds to your origin of replication. Later, MCM will come and bind. So MCM क्या है आपका? हेलिकेज है, ठीक है? Mini chromosome maintenance protein. So it comes and binds and it forms a pre-replication complex. So इसका मतलब हुआ है कि अब जाके आपका DNA replication शुरू होगा, ठीक है? Now the activation of MCM in S phase results from the action of the CDK2 cyclin E. It means MCM gets activated by the activity of your CDK2 cyclin E. So this is your ORC, this is your MCM. MCM is getting phosphorylated with the help of the CDK2 cyclin E. That is, it is getting activated. Okay. Now, the activation of CDK2 cyclin E will also result in the inhibition of APC ubiquitin ligase. Now, this is second protein is your DDK, which is going to phosphorylate your MCM proteins directly. Okay. So the moment APC is inhibited over here, the inhibition of APC leads to activation of a second protein kinase, that is DDK, which phosphorylates MCM proteins directly. So now what is happening is that the after phosphorylation, mini chromosome maintenance proteins, okay, that is the helicase, are getting activated. So once they get activated, it is going to open the DNA structure, right? And your replication is going to start. Is it clear? Any doubts?
No.
Okay. So here I think ORC is your origin recognition complex. MCM, as I mentioned, it is mini chromosome maintenance. CDK cyclin E. Okay, then DDK. What is DDK? DDK is your DBF4 dependent protein kinase. DBF4 dependent protein kinase. Okay. This is getting activated the moment APC is getting inhibited. Okay. Let's move ahead. Okay. Now the moment the helicase is initiated, okay, it is activated, the DNA replication is starting. ठीक है. अब क्या होगा, MCM प्रोटीन्स जो है आपके इस तरफ मूव होते रहेंगे जैसे जैसे आपका replication fork मूव करेगा. Right. Now the high activities of CDK during S, G2 and M phase, it prevents the MCM proteins from reassociating with the replication origin. So this also we know, right? See, there is a helicase and there is a helicase loader. So helicase loader क्या करता है? Only once it is going to load the helicase on the DNA so that the replication is happening only once per cell cycle. Okay. So the high activity of CDK during SG2M, it prevents the MCM proteins from reassociating with the replication origin. So pre-replication complex can only reform during the G1 when the CDK activity is high. Please remember, okay? Thus, once an origin becomes active during the S phase, replication at that origin cannot initiate again unless until mitosis hota hai and again the cell is coming back to your next cell cycle ka G1 phase. Clear? Is this much is clear? Like how we are regulating the DNA replication at the S phase? It is because of the high activity of CDK. So during S phase, we have got high activity of CDK, which is ensuring that your helicase loader is not loading the helicase on the DNA, which will result in the replication of the DNA only once per cell cycle. Okay. Now coming to the checkpoints. So cell proliferation is regulated not only with the growth factors but also by inhibiting its progression. यानी कि एक तो आपके पास growth factors रहेंगे कि check करने के लिए कि whether the cell is proper or not, and we also have the inhibitors to the cell cycle progression just to ensure that the DNA is not damaged. So we have got many checkpoints over here. So how we are going to respond to these checkpoints? By arresting the cell cycle at that stage. Okay, and making ensuring that the DNA is not damaged. अब ये कैसे पता चलेगा कि DNA damage है या नहीं है? So we have got these scanning proteins. So जैसे जैसे replication होता रहता है, although we know that the DNA polymerase has got this proofreading activity, still you know that after that region, let's say this portion of your DNA, ठीक है? So यहाँ से आपका replication हो गया है. So DNA polymerase ने check कर लिया था that there was no issue. But once the DNA polymerase goes further, there are chances that पीछे से कोई डैमेज हो जाए just because some ROS was present, reactive oxygen species was present, or any oxidative damage. It can be anything. Okay, then how to ensure that this is being corrected? So for that, at the S phase checkpoints, okay, before it enters to the G2 phase, it ensures that the proteins, okay, they scan the entire DNA and ensures that the DNA is not damaged. Okay, so these checkpoints, which are operative in G1S and G2 phases of the cell cycle, allow the time for the damage to be repaired before DNA replication or cell division is proceeding. Now, this cell cycle arrest is mediated by two protein kinase, ATR and ATM. So, nahi hoga. So, ATR is ataxia telangiecta RAD3, and this is the ataxia telangiecta mutated. So, when they are activated in response to the DNA damage. Guys, are you able to follow this much? Shruti? Okay. So now we'll talk about ATR and ATM. Okay, we can come back to this. Yes, it's always better to study with the help of the diagrams. So as we see, we have got the single stranded or the unreplicated DNA. It is going to, ATR is going to detect the damage happened to the single stranded DNA. Okay. ATM is going to detect the damage to the double strand break. Okay, इतना याद रहेगा. ATR is for your single stranded DNA damage. ATM is for your double stranded. Now, when ATR and ATM is getting activated, they then phosphorylate and activate check 1 and check 2. Okay, so they get the phosphorylated. Now, once they are phosphorylated, they inhibit the activity of the CDC25. Now, do you remember CDC25 kya karta tha? It's a phosphatase, right? Which was required to activate your cyclin dependent kinase 1 and 2 during the cell cycle, so that the cell from G1 to S phase it can transition. Now, since CDC25 इनएक्टिव हो गया है, तो वहां पे आपका फॉस्फर डीफॉस्फोराइलेशन नहीं होगा. इफ यू रिमेंबर इन द प्रीवियस लेक्चर, द टायरोसिन 15, थ्रियोनिन 14, right? So those phosphate groups are now not being removed. Hence, that MPF it will get it will remain inactive, and the cell gets arrested at that particular checkpoints. Then clear, Akshay? Is it clear like how ATR and ATM is functioning? Okay. So we'll just go through what reference is given. So ATR and ATM then activate the signaling pathway, which leads not only to cell cycle arrest, but also the activation of DNA repair. Okay. Now, as you know, if the DNA is not getting repaired, then the cell has to undergo the programmed cell death, that is apoptosis. The importance of this DNA damage response is emphasized by the fact that these proteins were initially identified because of the mutations in the gene coding ATM are responsible for the disease ataxia, which results in the defects in the nervous and the immune system, and eventually in the cancer. So right now also, guys, when we are working, okay, like in the cancer especially, देखा जाए तो maximum cases ATM mutation is resulting in the breast cancer. Like as far I have seen, like when I do the clinical trials analysis and all, what I've seen is that lung cancer is because of the EGFR mutation, breast cancer is because of the BRCA or HER2 mutations, and then also the ATM mutations. So always remember that it is not the one mutation which causes a cancer. At least minimum 5 major mutations are required. Then only a cell becomes mutated. Okay. Now, both AT and ATM are components of the protein complexes which recognizes the damage or the unreplicated DNA. So ATR is for the single stranded DNA, and the ATM is for the double stranded breaks. So once they activate the checkpoint kinases, check 1 and check 2 respectively, and they induce the cell cycle arrest by phosphorylating, inhibiting, or inducing the degradation of the CDC25. Okay, so as we saw CDC25, they are required to activate CDK2 and 1 by removing the inhibitory phosphorylations during the cell cycle. So this is the same figure, guys. If you remember, cyclin B CDK1 is there, right? So it is your maturation factor, maturation promoting factor MPF, right? So cyclin B CDK1 is there, so they get phosphorylation with the help of the V1. This is another protein kinase. So at threonine 161, threonine 14, tyrosine 15. Now CDC25 क्या करता है? It removes these two phosphate and hence make this complex active. Okay, which will go into the mitosis and results in the cell division. Okay, now what is happening is that the CDC25 is getting inactive by the check 1 and check 2 proteins and all. So क्या होगा उसके वजह से? This MPF is not getting activated. Hence, the cell cycle is getting arrested at that particular stage. Okay, either it will be G1 or S phase. So CDK2, if it is getting inhibited, then it is resulting in the cell cycle arrest G1 and S, and if it is CDK1, then it gets arrested in the G2. Okay, so summary क्या हुआ? CDC25 is inactivated. Okay, so your CDK2 is inactivated and hence it gets arrested in G1 and S phase. अगर CDK1 inactive होता है और inhibit होता है, then it is G2 phase. Okay, so that's how you arrest the cell in the particular cell cycle phases. So far so good, guys. Krishita able to follow?
Ma'am, the last point I didn't understand, CDK1 and CDK2 inhibition.
Acha, okay. See, this much is clear to you, like how check 1 and check 2 are getting activated.
Yes.
Okay, yeah, see, there is a single stranded damage, ATR gets activated. Double stranded DNA break is a ATM gets activated. So these ATR ATM is again the protein complex which activates your check 1 and check 2. Check 1 check 2 क्या है आपके? Checkpoint kinases, right? So check 1 ho ya check 2 ho, either it is because of the single stranded or double stranded. In both the cases, CDC25 is getting inactivated. CDC25 क्या करेगा? CDK2 और CDK1 inactive कर देगा. ठीक है? This is the meaning.
Okay.
Now we know that like in the G1 and S, CDK2 and cyclin E, they form the क्या बनते हैं? They form your complex, and hence if you inhibit CDK2, it results in the G1 and S phase arrest. यानी कि cell cycle उसी में रह जाएगा. Okay. Now, as you know, this is maturation promoting factor. ये कहाँ जा रहा है? If this is active, then the cell enters the mitosis. इसका मतलब क्या? Mitosis से पहले कौन सा phase है? G2 phase है. Agree? Right? हम ऐसे बनाते हैं ना? This is mitosis, G1, S and G2. If I am saying that active MPF then enters the cell cycle, sorry, into the mitosis, it means mitosis से तो पहले G2 हुआ. और अगर मैं CDK1 inactive कर रही हूँ, so it means I'm arresting the cell at the G2 phase of the cell cycle. Correct?
Okay, understood.
Yeah. Yeah, okay. And फिर हमें मालूम है after mitosis when the cell is entering the G1, there we have got our CDK4, 6, CDK2 which forms complex with the cyclin E. ये हमारा S phase में रहता है. The problem is that there is no clear cut division between the ऐसे G1, S या G2 या M. So that's why अगर CDK2 है, so we say that it might be G1 or S because G1 में CDK2 का concentration increase होने लगता है. Okay, so that's why we say arrest at G1 and S phase of the cell cycle if CDK2 is inhibiting, और अगर CDK1 है तो G2. Okay. Now coming to the P53 function. So I hope everyone knows what is P53 and क्यों इतना important है. Right? So there was this one interview question where people have asked like, why elephants, okay, till now never got cancer? Do you know this article about it? अभी वैसे तो कैंसर हमने कैट माउस नॉट माउस सॉरी माउस में तो होता है भी इंड्यूस. We never heard our animals or pet animals getting cancer. I mean, मैंने तो नहीं सुना है फिलहाल, but what I have seen is that, or what we have learned like through the scientific literature is that elephants are the only animals, okay, who can never ever get cancer in their entire life. ऐसा क्या होगा elephants के पास? Though they have got such a huge body, so the number of cells in elephants are, you know, many more times as compared to the human cells, the number of cells, right? So the chances of cancer happening in the case of the elephant should be more as compared to the humans, but scientists have proven that, sorry, they have proven that the elephants can never ever get cancer. even if you try to induce in them, it will still, not get spread in the elephants. क्या reason होगा? No idea. It is related to your p53 function only. So in the case of the humans, you have got only 2 copies on each chromosome of p53 or TP53 we say, right? Tumor suppressor protein. In the case of elephants, they have got more 4 to 8 copies of TP53. Okay, so there are many copies, multiple copies of TP53. And so they are highly regulated, and that's why the elephants can never ever get cancer. Okay, this is really very interesting article. If you guys get time, you can go through it. Okay, now coming to the p53 function. So in mammalian cells, they arrest the DNA damage. The arrest at the DNA damage checkpoint is also mediated by the action of additional protein known as p53, which is p53, which is phosphorylated by both ATM and CHEK2. So now we know that there is a DNA double-stranded DNA damages है, which is going to activate your CHEK2. CHEK2 क्या करता है आपका? As just now we have seen, CDK1 inhibit करता है, ठीक है? So आप G2 phase में रुक जाते हो. Now, in addition to that, CHEK2 and the ATM both are going to phosphorylate your p53. Now, phosphorylating p53 is making them active. And one more thing, guys, when you revise your cell cycle, make a table or something, because kuch phosphorylation is activating the protein and the other dephosphorylation is activating the protein. Okay, now which phosphorylation results in the activation, yeah, inactivation, that is very crucial to understand and to remember also. Okay, so here phosphorylation, abhi humne kya dekha tha? That CHEK1 and CHEK2, when they get phosphorylated, they are activated. But when they phosphorylate CDK1 and 2, they become inactive. ठीक है? तो ऐसे थोड़ा सा ध्यान रखना. Okay, so now when there is a double-stranded DNA damage, ATM, CHEK2, so they both together are going to phosphorylate your p53. Now this is what this will enter into the nucleus. It will go and bind and initiate the synthesis of your p21. Now what is p21? It is the CDK inhibitor. Okay, so it is going to inhibit the complex of your CDK2 cyclin E. So inhibition is happening then. Clear? Okay, is this much? Huh?
ATM itself also phosphorylates P53, right?
Yes, directly also.
Okay.
Okay, so now we know how p53 is ensuring that whenever there is a damage to the DNA, then by this process it is also arresting the cell. ठीक है? Clear? याद रहेगा? At least for now you have to remember, so that we can understand the concept. So I'll just say once again, there is a double-stranded DNA damage. ATM gets activated. It is going to phosphorylate or it is going to activate your CHEK2. Both ATM and CHEK2 are going to phosphorylate your p53. Okay, this gets activated and it helps in the synthesis of your p21. What is p21? It is your inhibition of your CDK2 cyclin E complex. Clear? अभी हमें मालूम है ना CDK2 cyclin E क्या काम करता है? It is present in the S phase. So we know the function. Okay. चलो. Now, what happens if there is no p53? या इसमें mutation हो गया है, ठीक है? Okay, so in most of the cancers, p53 तो mutated रहता ही है. So loss of p53 function as a result of these mutations, it will prevent the cell cycle arrest. It means what? No p53, no cell cycle arrest. Okay, so whenever there is a DNA damages है. So what will happen then? The damaged DNA is replicated and passed on to the daughter cells instead of being repaired. Okay. Now, the inheritance of damaged DNA results in increased frequency of mutation and hence leads to the cancer development. So, mutations in the p53 gene are among the most common genetic alterations in human cancer, which will illustrate that the critical importance of cell cycle regulation in life of multicellular organism. So, हमें पता है कोई भी चीज म्यूटेट होता है तो इवेंचुअली वो कैंसर में ही लीड होता है. Now, the next thing is about the mitosis. So this guys I'll be taking little fast because I believe you know how mitosis and meiosis are taking place, right? So we know what are the basics stages in the mitosis and the meiosis. The difference between the mitosis and meiosis, I won't be taking in detail. I'll just go through the content given, okay? Because it's very simple. We are learning from our childhood days. Okay, so M phase, it is the most important phase of in the cell cycle. So as we know here, the M phase, the replicated DNA, now this chromatin is going to condense so that it forms the chromosomes. Nuclear envelope will disappear, right? And the cytoskeleton will reorganize to form the mitotic spindle. So remember guys, we have discussed all this thing. So nuclear envelope breakdown means your lamin, okay? The lamins are getting what? Phosphorylated. So because of which the lamins are breaking and the nuclear envelope starts disappearing. Disappearing matlab they get fragmented actually. Okay. Now when we say cytoskeleton reorganizes to form the mitotic spindle, what does it mean? So inside the cell in the resting phase, we know that ये nucleus हो गया, यहाँ पे आपका centrosomes रहता है, right? So ये जो रहता है ये आपके ऐसे highway बनाते हैं. So that the motor proteins like kinesin, dynein, they can move on to these mitotic spindles, right? Vesicular trafficking, they help in that. Now once your cell is undergoing mitosis, then these mitotic spindles, so we know, right, these microtubules, they can disassemble यानी कि depolymerize हो जाएंगे and the centrosomes will divide. So what will happen now? दो जगह पे आपके सेंट्रोसोम्स हो गए हैं like this. There is no nucleus now. Centrosomes division has taken place. Now ये सारे आपके microtubules जो है disassembly या depolymerize हो जाएंगे and new polymerization of microtubules will start like this. So this also we have discussed that you have got the polar microtubules, astral microtubules, and the kinetochore microtubules onto in which kinetochore microtubules जो है जो आपके sister chromatids को bind करेंगे so that they can pull apart the chromosomes into the opposite poles, right? Then astral microtubules and the polar microtubules, they are going to help in the separation. We have done all this thing. Now, once the chromosome segregation is completed, the cytokinesis, the cell division will take place. Okay, so the sister chromatids then separate from each other and move to opposite poles of the spindle, followed by the formation of the daughter nuclei. So the moment the chromosomes which are there, okay, they are pulled apart, cell divides. So when the cell is dividing, by that time the nuclear envelope is also appearing. Did you get the scene over here, like what exactly is happening in the M phase? Yes, clear to everyone? Krishita, is it clear?
Yes, ma'am.
Yeah. Okay. So stages of the mitosis. So we know we have got the 4 stages: prophase, metaphase, anaphase, and telophase. The beginning of prophase is marked by the appearance of condensed chromosomes. This we can do with the diagram directly. I mean, I believe that you all know how the mitosis is taking place. Okay. Okay, so in the interphase, we know that the duplication is happening because in the interphase, S phase is there. So all the checkpoints, everything has been covered, and in the S phase, the DNA is getting duplicated. Now it is entering the M phase. M phase may be सबसे पहले prophase. So what do we see? That sister chromatids are formed, chromosomes have got condensed, centrosomes are duplicated, and the nuclear envelope starts disappearing. Okay, then what do we see in the prometaphase? no nucleus is present, and the microtubules or the spindle fibers, okay, they start attaching to the kinetochore present onto the chromosomes. Now in the metaphase, they align the chromosomes in one single plane, okay, to ensure that all the spindle fibers are attached to the correct chromosomes in a correct way. So they are correctly aligned. Then in the anaphase, what is happening? The spindle fibers are pulling apart the sister chromatids. Okay. Then at the telophase, what is happening? In the telophase, the cell is getting divided. Okay. So slowly, there will be a appearance of your nuclear envelope and the cell will get divided. So this is known as cytokinesis, and again the cell will enter the interphase of the cell cycle. Right. So this we know. Okay, can I go ahead with that, thinking that you guys know about the mitosis stages? Tanvi, सबको मालूम है, right? Okay. So now coming to the same thing, it just like what we have discussed now. The centrosomes separate and move to the opposite sides of the nucleus. See guys, please do also remember that the centrosomes are also getting duplicated during interphase, जो आपके S phase में. So as your DNA is getting replicated, of course the centrosomes are also getting replicated. ठीक है? So now they serve as the two poles of the mitotic spindle, which begins to form during the late prophase. So in the case of the higher eukaryotes, the prophase corresponds, the end of the prophase corresponds to the breakdown of the nuclear envelope. Then it enters prometaphase. Okay. Now in the prometaphase, the microtubules are attached to the condensed chromosomes. Now look at the figure over here. So these are the real live cell imaging. Again, interphase. You don't know what is happening. See, you can see the inside of the nucleus. Then early prophase, they start getting condensed. Late phase, you can see almost they got condensed and they start forming the chromosomes. Okay. Then in the prometaphase, you can see here cystochromatids are formed. Now they're aligning and now they're getting pulled apart. Right? Slowly, they are getting pulled apart and here totally divided. Clear? Now to get this red color fluorescence, they use PI, propium iodide. Blue color is because of DAPI, I have already mentioned. Green color is because of annexin 5HC. Red is propium iodide. थोड़ा सा pH चेंज करोगे तो उसका कलर रेड से ऑरेंज हो जाएगा. Okay. So the kinetochores or the cystochromatids are then oriented on the opposite sides of the chromosome. They attach to the microtubules emanating from the opposite poles of the spindle. So chromosome, they shuffle back and forth, and to make to ensure that the spindle fibers जो हैं जो correctly chromosomes के kinetochore को अच्छे से attach हैं, ठीक है? And then at the metaphase, they are aligned in the same single plate. Then there is a transition from metaphase to anaphase. Now this is possible only when the proteins which keeping your cystochromatids together are broken down. अगर ये चीज हम नहीं करेंगे, so what will happen? The cystochromatids are not able to be separated and then they cannot be taken into the opposite poles. So there are these certain proteins which actually ensure that the cystochromatids are held together. We'll come to that. So mitosis is going to end with the telophase, where during which nuclear reform and the chromosome starts decondensing. यानी कि वो फिर से आपका interphase में जाएगा. ठीक है? Now let's talk about the mitosis. Now अभी हमने just जो पढ़ा था, that is the overview of the mitosis. Now how these cyclins, the cell cycle regulators are ensuring that the mitosis is taking place properly. Okay. So now mitosis. So there are so many things happening. So we know that mitotic protein kinase, this is your MPF. Correct? Remember maturation promoting factor, MPF is there. So this is your cyclin B CDK1. अब this MPF क्या करता है? It is going to activate your aurora kinase, जो आपके पोलो लाइक काइनेस को एक्टिवेट करते हैं एंड इन रिटर्न वो CDK1 को एक्टिवेट करते हैं. ठीक है, विल अंडरस्टैंड स्लोली ये हो क्या रहा है. ओके, so mitosis involves dramatic changes in the multiple cellular components. It is triggered by the activation of CDK1 cyclin B protein kinase. Okay, now CDK1 cyclin B, they act as master regulator of the M phase transition. We know MPF, right? Why they are saying that it is a master regulator? Because first of all G2 से हमें M phase में जाना है. ठीक है, to ensure that all these the cell division is actually happening properly. ठीक है, so CDK1 cyclin B, they act as master regulator of the M phase transition by activating other mitotic protein kinases and phosphorylating some of them. अब देखो क्या होता है, जैसे MPF एक्टिवेट होता है, इट इज गोइंग टू रिजल्ट इन सो मेनी फंक्शंस. पहला, chromatin condensation. So here what will happen? Phosphorylation of condenses and cohesins. ठीक है, so they help in chromatin to become chromosomes. Nuclear envelope breakdown. So this already mentioned, phosphorylation of lamins. Okay, then lamin A, C जो रहते हैं वो आपके disassemble हो जाते हैं, because of which the nuclear envelope actually fragments. Then fragmentation of Golgi apparatus. Here the proteins which is related to the Golgi matrix, they get phosphorylated. Spindle formation, phosphorylation of centrosomes, kinetochore and the microtubule associated proteins, so that the microtubules or the spindle formation can take place so that they can bind to the sister chromosomes. Clear? So what are the four functions of getting activated, MPF getting activated? Chromatin condensation, phosphorylating your condensins and the cohesins, nuclear envelope breakdown, phosphorylating lamins, fragmentation of Golgi apparatus, that is phosphorylation of your Golgi matrix proteins, and the spindle formation. Okay, so phosphorylation of centrosomes. So what do we see here? Each step, each 4 cases, phosphorylation is only happening. Okay. Now, mitotic protein kinase, aurora kinase, we have got aurora A and B, and polo-like kinases, they are activated coordinately with the CDK1 signal.
So guys, till now we are testing
1 second. Okay, I'll just say once again. So the protein kinases, okay, aurora kinase A and B and your polo-like kinase, they are actually getting activated along with your CDK1 signal, okay, which is the entry to the M phase. Clear? अभी देखो हम धीरे धीरे जाते हैं. So we have got your CDK1. Okay, this is activating your aurora A and B, aurora kinase and polo-like kinase family. So when they get activated, इसका मतलब क्या? We are entering into the M phase. Okay, so aurora and polo-like kinases, they function with CDK1 in a positive feedback. इसका मतलब क्या? ये इसको activate कर रहा है, ये इसको activate कर रहा है, ये इसको activate कर रहा है. So this is like, positive feedback, like keep on activating. ठीक है? So the condensation of interface chromatin to form the chromosomes of mitotic cell is a key event in the mitosis. Highly condensed chromatin cannot be transcribed, so most of the time the transcription ceases as the chromatin condensation takes place. So obviously बात है ना, that whenever the chromatin is getting condensed, that time we don't need any transcription or any kind of replication taking place, right? So since they are highly condensed, so none of the transcription will be taking place, irrespective of the region, whether it is a euchromatin or a heterochromatin. Guys, is this slide clear? Is this part is clear? That cyclin B in CDK1, this is your MPF. It is activating aurora kinase. Aurora kinase activates polo-like kinase, and this is going to activate your CDK1. So it's a positive feedback loop. Clear?
Yes, ma'am.
Okay. Now chromatin condensation. अब यहाँ से थोड़ा और ज्यादा ध्यान देना है. So chromatin condensation is because of the protein's condensance. Easy to remember, right? Which are the members of the SMC, structural maintenance of chromatin, which play important role in the eukaryotic chromosomes organization. Now, both condensance as well as cohescence, they contribute in the chromosome segregation during mitosis. So here we have got two proteins, condensance and the cohescence. Okay, now cohescence bind to DNA in S phase, maintain the linkage between the sister chromatids following the DNA replications. Okay, so we have got here two sister chromatids and both are linked by the cohesence. And as the cell enters the M phase, condensins are activated as a result of the phosphorylation by both CDK1 and Aurora B kinase. So just now we saw. So MPF activating Aurora. Okay, Aurora क्या करेगा? आपका condensin activate करेगा. ठीक है? Now, condensins will then replace the cohesins along most of the length of the chromosome, so that the sister chromatins remain linked only at the centromere. अभी अगर आप देखो, if you know, I don't know whether someone has told you or not, your sister chromatids are like this. Centrosome में आपका centromere में they are connected, right? Cohesins क्या करता है? It forms like a ऐसे connections like this, so that the sister chromatins are held together. Okay, now as just we saw CDK1 getting activated, Aurora B kinase getting activated, your condensins are getting activated. Condensins क्या करेंगे? इस cohesins को remove कर देंगे. So क्या होगा? Only at the centromere it is attached. ठीक है? हाँ, इतना clear है? And of course condensins are helping in the condensation of the chromatin to form a complete chromosomes. Is this much clear? The takeaway message of this slide? Okay, yeah, here it is. See how beautifully they have shown. So when your DNA is getting condensed, okay, chromosome se ban raha hai. So already, within nucleosome, solenoid structure and all, it has formed. So then they start forming the loop-like structure like this. Aise, ye. Okay, so this is your DNA. Now what happens is that these are the cohesins. So both the sister chromatins are, attached like this. Then centromere is there. Condensins क्या कर रहा है? It is helping in the formation of the DNA loop, so that again and again इतने लंबे के जगह you get it a condensed short structure. Now your CDK1, cyclin B, Aurora B, polo-like kinase are getting activated. So what is happening? Only at the centromere it is attached, and rest all हमने free कर दिया है. Clear? Is this much is clear? Yeah. Okay. ये था आपका पहला. That is when where we have talked about the condensins or the chromosome, or the chromatin condensation. Second क्या है? Breakdown of nuclear envelope. There were 4, right? When MPF is getting activated, there were 4 functions. तो पहला हो गया आपको chromatin condensation. दूसरा हो गया breakdown of nuclear envelope. So breakdown of nuclear envelope means nuclear membrane is getting fragmented, NPC are getting dissociated, and nuclear lamina is getting depolymerization, that is by the phosphorylation, right? So depolymerization of the nuclear lamina is also from the phosphorylation of the lamins by the CDK1 cyclin B. So phosphorylation causes lamin filaments to break down into the individual lamin dimers, and hence we say that the nuclear envelope is disappeared. Okay, so we have got the nuclear membranes, NPCs, nuclear lamina niche hai. Now once they start getting phosphorylated, they lose their association with each other. So they start forming the dimer and they get separated, because of which now the nuclear membrane, which is जो आपके lamins के support में था, since lamins ही नहीं है तो क्या होगा? When you don't have the support, what happens? The wall fall off, right? So that's why the nuclear membrane starts fragmenting. Third क्या था? phosphorylation of the Golgi apparatus proteins, right? So the Golgi apparatus and the ER starts fragmenting which may either be absorbed into the ER or distributed directly to the daughter cells at the cytokinesis. And that's how, both the daughter cells are having chota portions of the Golgi apparatus and the ER. And as the cell is growing, the Golgi apparatus as well as the ER, they start also growing. Okay. So breakdown of the Golgi apparatus into vesicle is mediated by phosphorylation of multiple Golgi matrix proteins by the CDK1 and the polo-like kinases. Okay. And the spindle fiber formation microtubules ye bhi hame pata hai. So breakdown of the nuclear envelope then allows these spindle microtubules to attach to chromosomes at their kinetochores. So this initiates the process of chromosome movement, which will characterize your pro-metaphase. So guys, so far so good. What happens when MPF is getting activated? It is activating aurora kinase, polo-like kinase, right? And then these four functions are happening: chromatin condensation, nuclear fragmentation, then phosphorylation of the Golgi proteins, and the last spindle fiber formation. Okay? Clear? Now, the proteins then assembled at the kinetochore include aurora B kinase involved in the interactions of chromosomes with the microtubules. Okay. Now, microtubules attached to the chromosomes. Microtubule motor directs the movement of the chromosome. So guys, अभी हम यहाँ पे वो बातें नहीं discuss करेंगे which we have already discussed. If you remember in the microtubule we have talked about like how these kinesin and dynein proteins, they carry the chromosomes, sister chromatins, and they walk onto the microtubules and results in the movement of the sister chromatids in the opposite directions, right? So here I'm not gonna explain that same thing again and again. So these action of the proteins, okay, which draws the chromosomes towards the centrosome, is opposed by the plus and directed motor proteins, and they all astral and interpolar microtubules, they will help in the movement of these motor proteins so that they can bring the sister chromatids in the opposite direction. Eventually what is happening, they are making sure that यहाँ से separation शुरू हो जाए आपका, ठीक है, और one copy of the genome comes here and another copy goes in that direction. Yeah. So guys, that's all for today. Any doubts anywhere you face and you didn't understand, so I can explain that. So we can just summarize now what we have discussed today. So we discussed about the DNA replication ka regulation. So where we talked about ATR, we talked about ATM. ATR is for your single stranded DNA damage. ATM is your for double stranded DNA damage. They activates your check 1 and check 2. What are these? They are your checkpoint kinases, right? Then they activate, sorry, not activate, they activate CDC25, sorry, inhibit CDC25, which is a phosphatase, correct? A phosphatase is going, when this is inhibited, this is going to inhibit the activity of your CDK2 and CDK1. So CDK1 रहा तो G2 phase हो गया, CDK2 रहा तो आपका G1 या S phase cell cycle arrest होगा. Clear? Then we also talked about the action of your P53, how it is regulating. If there's a DNA damage, it is getting phosphorylated. See, DNA damage raha to aapka ATM gets activated, right? ATM is going to activate your P53 by phosphorylating. Aapka check one yahan pe hai. Now P53 once gets phosphorylated, it goes and binds to your DNA region where it is going to induce the transcription of your P21. P21 kya hai? Inhibitor of your CDK1 and cyclin E, right? So that's how also you can arrest the cell cycle. Guys, anything else? Okay, no questions. It means everything is clear.